Welcome back to the third video in our series on cell signaling. Last time we talked about some general types of cell signaling, as what we're going to do today is start looking at some specifics of the signaling process. Now don't forget, I'm talking in general terms here. There's so much diversity in signaling pathways that we would have to get into lots and lots of different examples to give you some concrete ideas to take away. So we're talking about general ideas here, and so when I talk about the three stages of signaling here, you will see variations of this when you start looking at specific examples when you take cell biology or physiology or some advanced level biology classes. So we break signaling down into three stages: receptor activation, signal transduction, and cellular response. And so we'll talk about each of these in turn, and then we will just look very briefly again at receptor activation at the end. So let's start over here on the left with receptor activation. A cell that's going to respond to a signal from another cell must receive the information it's being sent, and that's done using a receptor. So these are proteins. We're going to start looking at these on the cell surface, and towards the end we'll start looking at them, or we'll finish up looking at them in the cytosol, in the cytoplasm. So we've got a receptor here, and it's on the cell surface. Here's our membrane, and here's our receptor. That's going to be the molecule which receives the information. And here's the signaling molecule, and that is going to have the information that's being sent by another cell. So the receptor is either inactive down here at the bottom or active. It's got two states, inactive or active. When the ligand, the information containing molecule from another cell, arrives at this cell and binds to the receptor gets turned on, and now it's activated, which means that it's going to send the information that it's received in that ligand into the cytoplasm. And so what we see is a structural change inside the cell in the receptor protein when the receptor binds to ligand. So that's the first stage, receiving information. The second thing that has to happen is the information is going to get from the cell membrane to wherever the cell is going to have the appropriate response. So this is between the reception of the information and the response, is this transmission of information through the cell, usually through the cytoplasm, sometimes into the nucleus. And this is called signal transduction. And so generally what's going to happen here is that the activated receptor will bind to a cytoplasmic protein and turn that protein on, and then that protein will bind to another cytoplasmic protein and turn that one on, and this keeps happening until the information is eventually transmitted through the cytosol. Now these pathways can branch, and so maybe this purple protein in the middle here is actually going to turn on several other signal transduction proteins, not just this yellow one here. So there's going to be a lot of branching and talking between these pathways, and that's what leads to the complexity. So once the information gets to a point in the cell where there can be a response, then there are going to be general responses, things like cell death, cell growth, cell division, differentiation, and those responses are facilitated, or they're brought about, I guess, by changes in things like metabolic enzymes in cells. So if we've got to divide the cell or we've got to grow, we've got to make more ATP to support that metabolic behavior, and so maybe enzymes involved in glycolysis, the oxidation of pyruvate, and the Krebs cycle, for example, maybe they will be turned on by a certain signal which tells the cell to grow. If the cell is going to divide, we need to rearrange the cytoskeleton, and so we may see changes in structural proteins like the microtubules and the microfilaments. And many cellular responses are dependent on changes in gene expression. So under one set of physiological conditions, you'll see certain genes being expressed, but then in receipt of information from a neighboring cell, the cell will change the genes which are being expressed, and therefore its structure and function can change. For example, if we wanted to differentiate a cell into a particular cell type. So we have general responses like growth, death, division, differentiation, and those changes are brought about by more finer responses or more higher resolution responses, where there's changes in enzyme activity, structural proteins, or transcription factors changing gene expression. Let's just go back and review the beginning of this receptor activation. Here comes our ligand here in red. So here is our ligand. That's the information containing molecule. It's going to bind into this site on the inactive receptor, and that means that this structural piece of the protein is going to change once the ligand binds, and that will start the signal transduction pathway. Now, you don't always want these things to be on. Think about it like a switch. The receptor over here is off, and here it is on. You don't want these things always on. You sometimes need to turn them off. So the receptor will sometimes let go of the ligand, and the ligand will then dissociate. It'll unbind from the receptor. When that happens, we go back to the receptor being in the off state, and we've turned the signaling pathway in the cell off. So we can switch them on, we can switch them off. Okay, those are the 3 stages of cell signaling. I'm going to wrap up here, and next time we'll start getting into some more details of other parts of the transduction pathway and the responses. So I'll see you soon.